uh, resource person. The guest speaker for this second session is nothing Dr. Atya Kapley. She is also working as a chief scientist and head of environmental biotechnology division. Madam, am I audible to you?
Yes, I can hear you.
Yeah. Dr. Atya Kapley, madam, is working as the chief scientist and head of environmental biotechnology division of National Environmental Engineering Research Institution. And the topic of her presentation is harnessing the microbial community intelligence for the environmental protection. Now, again, I would like to request Dr. Vishal to please introduce the today's guest speaker.
Yes,
Dr.
sir.
Vishal, please.
Am
Dr.
I audible,
Vishal?
sir?
Yeah, you are audible. Please continue. Please proceed for the brief introduction of the madam.
Good afternoon, one and all. I am Dr. Vishal Daulatkar, Assistant Professor in History, Art Commerce College, Ralegaon. It's an immune pleasure and privilege for me to introduce to the resource person to refresher course on environmental science. Thank you, Dr. Praveen Meshram, sir, for shouldering this responsibility. On behalf of the whole team of UGC, HRDC, Nagpur University, and all participants, I welcome Dr. Atya Kapley ma'am. Dr. Atya Kapley ma'am, an esteemed scientist and visionary leader in the field of environmental biotechnology and genomics. With a current position as a chief scientist and head of the Environmental Biotechnology and Genomics Division, CSIR, NIRI, Nagpur. Dr. Kapley has been a driving force behind numerous groundbreaking research projects. Here are some key highlights that showcase her remarkable contribution. Extensive professional experience. Dr. Kapley boasts an impressive research experience spanning over 30 years. Throughout her career, she has adeptly combined scientific and administrative skills, successfully leading several national and international projects. Focus on two critical domains. Dr. Kapley works emphasis two essential areas: generating knowledge through fundamental research and actively applying science for betterment of society. She has worked on developing monitoring protocols and using genomics tool to address issues related to sludge and wastewater, with a particular focus on bioremediation for degraded land and polluted niches. Pioneering research and expertise. Her expertise has led her to spread flagship project aimed at remediating contaminated sites, particularly addressing the long-standing problem of untreated sewage in India. Her leadership in multi-expertise team combining microbiology, genomics, engineering, and computational science has been instrumental in the success of the projects. International collaboration and empowerment. Dr. Kapley has instrumental in establishing network project with various national and international expertise, fostering collaboration with countries like Finland, Estonia, Portugal, the UK, the Netherlands, China, France, Singapore, Denmark, and Israel. Moreover, she actively works to empower women in science as the Vice President of Organization for Women in Science for the Developing World, Asia Pacific region. Her outstanding contribution have been recognized with the numerous prestigious awards, including the Young Scientist Award from the Association of Microbiologists of India and the CSIR Technology Award. Dr. Kapley is a fellow of the Maharashtra Academy of Science and holds membership in several professional groups. Dr. Kapley's dedication to her field extends to her active participation in various committees and advisory groups related to environmental biotechnology and capacity building in taxonomy. Dr. Kapley research focus on monitoring environmental pollutants and studying their impacts on ecosystem, leading to innovate remediation solution. Her multidisciplinary approach combining microbiology, engineering, molecular tools, and bioinformatics has proven instrumental in addressing environmental contamination. Dr. Kapley's global collaboration have led the publications of 126 research paper in Science Journal. She has also monitored and supervised several PhD students with 10 awards and 5 ongoing. In conclusion, Dr. Kapley stands as an explanatory scientist and leader, making significant contribution to the field of environmental biotechnology and genomics. Her dedication to bridging the gap between scientific research and social impact, as well as her commitment to empowering women in science, make her an inspiration to all aspiring researchers and professionals in the field. May I request Dr. Atya Kapley ma'am to please enlighten us with valuable insight on harnessing microbial community intelligence for environmental remediation. Dr. Kapley ma'am, over to you.
Thank you, sir, for that very kind and a little detailed introduction. I'm really happy to be a resource person on this, esteemed group. And I would like to thank Professor Meshram for inviting me as a resource person. So just give me a second and I will share my presentation. Can you all see my slides?
Yes, ma'am.
Okay, I'm just making it full
Yes,
screen
ma'am.
mode. I hope you can see it now also in a full screen mode.
Yes, ma'am. Yes, ma'am.
Okay, thank you. So I'm going to talk about how my group deals with environmental pollution. I don't need to tell anybody, that the environment, at least, in developing countries and more so for us very specifically, all the environmental parameters are at an all-time low. We have managed to pollute all our rivers. We have degraded land everywhere. You see the fury of the nature because in response to the degraded land, we have been just seeing the last month of all the horror stories. Of course, it also contributes in to climate change. There is something that everybody can contribute to remediate this environmental pollution. Me and my group at NIRI, we look at the potential of the microbes and more specifically the bacteria in combating the environmental pollution. So the theme today, which I was given to understand, is to understand the various aspects of pollution prevention to attain the goals of sustainability. So my work mainly focuses exactly on this. But before I start, I thought I'll give you a little introduction to NEERI. I'm not so sure whether this participants are all from within Nagpur or also from outside. So for those of you who are not in Nagpur, you would have heard of NEERI, I hope. It is the only national institute which is dealing with the environment from the CSIR, which is the Council of Scientific and Industrial Research. There are 37 labs, and NEERI is the only one which is dedicated to environment. We are based at Nagpur, and the headquarters are here. And NEERI has different zonal centers in New Delhi, Mumbai, Hyderabad, Chennai, and Kolkata. And all these red pins that you can see are all the places where we have gone and taken up projects and delivered on this subject of environmental remediation. We are divided into different groups. So because, you know, the environment is a very complex entity, it comprises of different components. And so for each component, we have a different division. And like sir introduced me, I head the environmental biotechnology and the genomics division at NEERI. So let us start with what exactly creates the problems. You know, why has environment deteriorated so much? I just had joined in a little earlier than the start of my session, so I could hear Dr. Ramesh Kumar's discussion points, and there was a lot of, you know, things, questions being asked to him about plastics. You know, it's not that the plastic invention was bad. Plastic has come to many uses. In fact, many uses, but it is the use of the community. You know, the carelessness, the sheer wastage, you know, not making it a sustainable thing that has created this mountain of problems, just like we have done for everything else. So just take a look. What we do is we take in inputs from nature, you know, water, food, land, energy, all this we take in from nature. And, you know, what do we give to them? We don't, you know, segregate our waste. We don't manage. We have just managed to pollute, like I said, all our wastewaters. Wherever you go within the country, you can see heaps of solid waste. There's hardly any place. I think with the exception of Indore in the country, there is no management of solid waste going on at the scale that it is generated. We are doing work. It's not that, you know, the government just sits and the policymakers are not doing anything. They are doing, but it is not enough for the amount of waste that is being generated. And what is happening? We are cutting down so many forests. We are ruining ecosystems. So obviously there's going to be biodiversity loss. Biodiversity loss means your ecosystem services are being hampered. And, you know, climate change, I don't have to say anything about it. So you can see that the graph is flowing only in one direction. And, you know, no unilateral graph like this, you know, unidirectional can ever be a sustainable option. We have to bring this to a full circle. We need to remediate water so that there is no pressure on the natural resources. We are getting food, but agricultural land also gives you so many other problems. We are misusing pesticides. We are cutting down the land of, you know, other than the Homo sapiens, there is no respect for the rest of the species of the world, very sadly. So we need to, you know, make this into a circular path, and then only we can. So I'm going to be focusing on two aspects. One is degraded land and the other is contaminated water. So normally when we say degraded land, most of the research that you will see is on contaminated soil. In NEERI, we are working on contaminated soil, that is pesticide contaminated soil, oil contaminated soil. We are also working on, fly ash, moin spoil dumps. But there are other different kinds of degraded land. When you have dry land, when your deserts are expanding, that is also a land degradation. Logging is also a degradation. You have nutrient imbalance. There's so much of salinity all across the Gujarat coast and, the Kutch regions. You have so much of saline soil. So it's very difficult to, actually take groundwater for anything, your agriculture, your drinking water purposes. And of course, all the hilly regions, riparian zones, there is continual soil erosion. Some of those reasons are because we are cutting down, to make new roads. We are not taking care of the slopes. So which is why the soil keeps also getting eroded. It is also a natural process, but what we are doing is enhancing that process, thousandfold, which is why we see disasters like this. Of course, the major drivers are us. When we say anthropogenic activity, it means we, the Homo sapiens, who are totally destroying the ecosystem. We have urban encroachment. I will show you some slides, of how urban encroachment is changing the landscape, So the improper land use, what we call. And of course, soil biodiversity is going down simply because of the chemicals that we are piling onto it. So this is just a very quick glimpse of a few projects that I have done on pesticide contaminated soil. So we chose atrazine as there are many number of chemicals that is done in Niri. I'm just giving you an example. So we looked at atrazine because even though it is banned in Europe for the last 15 years or so, it is still used in India. It is a herbicide. Basically, it is used to control weeds. And so it is very widely used in sugarcane and corn. These are the two main crops where it is used. This atrazine has been proven to, constant exposure to atrazine has shown that it causes sex reversal in frogs. This is a level of the toxicity behind this compound. So what we did was we went to the fields where they are using atrazine. We got back the soil. We isolated bacteria. We found bacteria there that are able to degrade atrazine. And we isolated those bacteria. We made a consortium of those bacteria. And, although you get it from the soil itself, but the numbers are very low. So we multiply them and put them back into the soil. So the, atrazine content in the soil is speeded up. The biodegradation is speeded up. So we have done the genomic analysis. We have delineated all the genes that you can see in this pathway on the left side of the figure. And we have shown that different bacteria contain different, pathways. And they are all able to use atrazine as a nitrogen source. So this is one kind of work. We also had a collaboration with Finland on this. And even though atrazine has been banned in Europe for the last 15 years, in Finland, because of their climatic conditions, so cold, the atrazine in the ground and in some groundwater still remains. Luckily for us, we live in a tropical country where the temperatures are good and biodegradation, aeration, and most of the country has good temperature cycles. So our biodegradations are a little faster. There's another project that we are trying to take to the field, which is crude oil contamination. So what we are doing here is because we have oily sludge, which is generated in all these oil refinery. Crude has to be processed into different versions of what we use. And during that process, a lot of oily sludge is generated and it is deposited at ONGC like this, like the pictures you can see at the bottom. What we are doing is trying to remediate this kind of waste so that, the fields nearby are not contaminated. the oil, and oil is not just oil that we use from petroleum. When you're extracting, there are so many priority pollutants, the 16 phs are there that are toxic, and if they leach into the groundwater, so you're creating a health hazard for the whole region. So we are working right now, and we are actually going to field near the ONGC assets at Ankleshwar and in Assam to apply our technology to treat this kind of soil. So in the papers recently, you have read that, fly ash, because of these rains, there was, a breach in the dike, and all the fly ash, which with the rain, which became a slurry, just went into agricultural lands and, has created so much of toxic problems, ruined all those agricultural fields. So one scientist from my group, Dr. Lal Singh, he has developed a technology where he has created bamboo forests. If you can see here, this is an absolute fly ash dump. And from that dump, through different kind of treatments, he has planted, which of course includes fungi and bacteria. And he has created this kind of a forest. So you can see here that, the fly ash has been converted into a kind of a soil. And this completely, reverses the toxicity of this fly ash. So such kind of work is extremely important to take into the environment. We also create, different remediation protocols for different needs. So when you have an industry which is generating wastewater, we have a treatment system which is called the high rate transpiration system. It involves, using of plants. So you can see we channel the water here. And so these plants, because they have a very high rate of transpiration, they take up that water. And so the industrial wastewater is used for these plants to grow, and also prevents this toxic water from reaching the environment, polluting our lakes. So this is in force in many different industrial premises. It is also done in my lab by two scientists, Dr. Karthikeyan and Dr. Towley. So basically, if I want to summarize what we do for the soil, I would say that we make waste-specific consortia. Because say atrazine will require a different kind of a consortia. Endosulfan, it is banned now in India, but traces are still there. DDT is banned for many years, but it is used not as an insecticide, but in some other purposes. So there are many xenobiotics. So we look at a consortia that can target different antibiotics. So obviously you need different bacteria for different xenobiotics. So we look at soil amendments. When you have a microbial diversity, natural microbial diversity of the soil, and when you keep on adding some kind of a pollutant to it, whether it is oil, whether it is, agriculture, or whether it is just the topsoil getting eroded with the rain, constantly. So there is no microbial diversity there that supports that soil to grow anything. So we look how to do soil amendments, and we use different bacteria that can, do nitrogen fixation, bring back the fertility of that soil. We have worked on, plantation corridors on highways, so to absorb all, the gases, etc., from the vehicles, to create a different ecosystem along the highways. We have looked at reclamation of wastewater-laden soil, like I showed you in the last slides. And doing all this, we have developed an expertise in microbial diversity, which helps us to take on different projects that can be aimed at not only at degraded land, but also at water. So I'd like to show you, some pictures where there is nothing that a scientist or a microbiologist can do. It is just the policy. It is people like you and me who are creating these kind of problems. Now, this is a Google Earth image of the Musi River in Hyderabad. This black line that you can see is the river. Now, water looks black in when you view it from very high. So now you look at this river, and all these white dots and squares that you can see is all the built up. So around the river, even the year 2000, not so far back, there is some kind of greenery. What you say, the embankment of the river, the catchment area around the water bodies, that should be left untouched, because those are what manage the river. When the river is in flood, or, there are so many other ecosystem services that these spaces do. Those vacant spaces are not for commercial, use. They are meant to give stability to that river. But take a look at what has happened in just 2017. You have built up right up to the river, and now the black has changed to gray, which means that there is so much of, some, what should I say, suspended solids in it, which are obviously they are coming from sewage, but you can see the amount of damage that has been done to a natural community. And this is just 17 years span. In 2000, all of us were there, So, and just look at how the population is growing. So this is the kind of effect we are having on the natural ecosystem. Obviously now you're going to have hazards of floods because the river doesn't have its embankment anymore. And if you're going to be building this in the hilly regions, then obviously you're going to be facing landslides. There is also like another kind of degraded land, like I said, desertification. A desertification atlas has been initiated by ISRO. And, when we look at what are the causes, we find that majority of the causes are, of course, man-made. We have overgrazing. Wherever there are grasslands, if there is overgrazing, then there's going to be no grass left. So different farmland practices like burning, when the stubble burning issue we read every winter. Deforestation, all of you, if you're reading the paper, you know that from Nagpur itself, how many areas the trees are being cut down for urban development. I'm not saying at any point that urban development should not happen. Of course, it should happen, but there has to be a balance with nature. There is a critical threshold that you should not cross. You can find, ways and means, but it is this utter disregard for that critical line that is causing so many, issues that we are actually facing today. And of course, natural causes are there. If there is a system that's going to have a particular life. But like I said earlier, we are, increasing that threshold by more than 1,000-fold. So what happens, we don't take into account what are the impacts of this. They are obviously going to be very high economic impacts. There's going to be a threat to agriculture if your land is degraded. You're going to increase poverty, when the population is so high and you're in agricultural land is, what should I say, degraded. How will you feed the population? Overall, there are very heavy impacts. The forests give so many ecosystem services. If you're cutting them down, you're going to face at some point of time an impact on your economy. And of course, there's social impacts. Like I said, poverty is increasing. There will be famine. You're going to have social conflicts. You're going to have migrations. Because you don't have a particular environment, there will be different migrations, food security issues, and there are also cultural issues. Nobody documents the cultural issues, but cultural issues are also there. Actually, my team makes the environmental status report for the Nagpur Municipal Corporation. And we studied the urban, green spaces. So if the Ministry of Urban, Affairs tells that you have to say 20 hectare per, this kind of a population, in Nagpur, we are able to give only 2. So you see, there is cultural. Would you not like to, live in an area which has a park nearby where your children can go and play, where you can take your dog for a walk? There are no such spaces left in the city. You can hardly count them on your fingertips. When I was a child, we had a nice place to play with, all the children of same age used to gather, we used to play. And, our parents were confident that my children are safe. But today, it's very difficult for us to give our children that kind. So we're already, taking away something from the growth and development of our children. So it's not that, when you hamper the environment, that it stays with the environment. You can say, "Okay, today there is rain and today it's flood, tomorrow it will go away and we'll come back." No, you're impacting so many different spheres of life that you are not looking into. So what can we do? So, we need to first understand what are our natural resources. We need to document them. Of course, there are survey of India and different DRDO, ISRO, all of them have it. But that has to be made like public. The people need to understand. Unless, my philosophy is that unless, people are involved in environmental sustainability solutions, it's not going to happen at all. And I will show you later why I say that, in some slides of my water. So isolated policies, of course, they really don't work. But sadly, one department works. It's not only the government departments, anywhere if you see, this one has one issue and some other department has another issue. They all actually combine to make a holistic program, but they will always, work separately. We have to integrate our land. So land is a different issue, water is a different issue. But ultimately, a farmer needs land and water. So unless you integrate these policies, it's really going to be. I was told when we were conducting a survey and we asked some farmers, and we said, oh, you're getting so much of support and things, and why is still your productivity not being? And they said that yes, madam. So what happens is, we are given electricity and many times free electricity. But say from Monday to Wednesday, it is there in the morning, and from Thursday to Sunday, it comes in the night. Now tell me which plants can be watered in the night. So these kind of policies, they need to be in tandem to what is actually required. And of course, that's policy level matters, and that is not something what a scientist can do other than give suggestions. But we will look at now what we can do. We can protect soil erosion. If you just have a healthy tree cover, soil is automatically rooted. If you can't have space for trees, you can just put weeds. Even weeds on the slope and grass are going to hold that soil. So this is something that all of us, can recommend, can take up small projects. Our students should be taught, because they are the ones who are going to live in this future. We have caused so much of damage, but if we want to recover and explain to them that environmental sustainability is something that can be achieved with patience and with a little bit of compromise. Oh, sorry. So I'll come to water. This is the pictures that you see. Of course, I got them from the internet. But I'm sure that there would not be even one in this audience who has not seen something like this. So this is the condition of our water. India is really blessed to be, have so much of natural resource when it comes to water. We have so many rivers, we have so many natural water bodies. But what have we done to all of them? This is not something that, any policymaker or any government can work with. This is made by people like you and me. Yes, here, this you can see these influence coming into, this is industrial. Yes, now this is something that should be stopped, and we make so many agitations and things like that. The industry should stop putting that in the water, blah, blah, blah. But this is not something that any industry has done. This is not something that, any government has done. This is people like you and me who have created this. So when we do not respect our environment, can we really expect that we have safe, clean drinking water? Can we really expect that we are going to have a nice, beautiful place, clean water? We have done this to every possible water body, and that is just, based on citizens. So there was a time, when we could drink water from the tap. When I was in school, I never carried a water bottle because there was a tank, in the middle of the school ground, and all the students drank water from that tap. But my daughter did not have that kind of a privilege because we, the citizens, contaminated all the water that could be. And it's not only about water contamination. Our reality is that we are in a water crisis. For a country that has abundant rivers, for a country that has so many lakes, how many cities are known as lake cities in our country? For a country that has so many abundant forests, we still have water crisis. We still get water from tankers in many cities. We don't have enough of water to supply on a daily basis. And so those who have water supply, 24/7, they are very lucky. I don't know how much of that percentage is there for India. There are so many villages that still need to be supplied water in tankers. So India is facing a really high water crisis. But everybody expects that somebody else will solve this problem. Nobody thinks that, what we can. So, it is globally, it is not only India, everywhere you have, most of the countries, you have a proper water crisis. So you see the blue, these are the people who are managing their water. So look at China also, also managing its water. And we are, here we are. I have no words. So what we do in our lab is try to remediate the water environment. So we are a group of biologists. So we have the expertise of microbiology, of botany, of zoology, of genomics. And so we combine all these subjects and, take help of our engineering faculty, which is also there in NEERI, and try to do a remediation of the environment. So, what is happening is that water, most of our fresh water is going into, agriculture and some into industry. And 80% of water that is supplied to you is, converted into sewage, which just goes down the drain and contaminates the nearest river or water body. So if we can treat that, if we can use wastewater treatment and reuse that, water, so then we can use that treated water into agriculture and industry. So fresh water reserves are conserved for our drinking purposes, But the warning here is that you cannot do this half-heartedly. If you're going to do that, you're just introducing more pathogens into your agriculture, and somewhere through that food, it is going to reach us, again. So wastewater management is, I would say, a really top priority that needs to be taken really very seriously. And bioremediation is not a new concept. Even in 600 BCs, it is said that the Romans, they pumped out all their sewage into somewhere far away from the city. And they had, like we have our normal sewage treatment plants, they had some version of that. And they respected water so much that they even installed one goddess there, and she was called, the goddess of the sewers, which means that she treats that waste water and, makes it less polluted. Although, at that time, the population was not like what it was today, so they did not reuse that water. But this is a story that, we found. So the problem in our country today is that we do not have the facility which to deal with all the wastewater that we are generating. If we are looking only at, say class 1 or the urban area, I'm not even counting the small towns and the villages in this estimate. If we are creating 1x amount of wastewater, we are having only the capacity to treat 40% of the wastewater that is generated in our homes, in our offices, and the remaining 60% is just flowing down the drains, untreated, and reaching the nearest lake or river. Ultimately, it goes into the river. If those of you living in Nagpur should go once and, look at the night talab. It used to be a talab, which means a lake. But now it is a holding area for the sewage that is coming in from that area. And when that lake water fills, they open the gates and it goes and joins the Pench River. So this is the condition, what we have done to our lakes. So there are two ways, that you can treat this. Either a centralized treatment, which, should be done by the city planning, basically. But it requires a lot of, expenditure. It requires not only the creation of your sewage treatment plants, it requires an absolutely different plumbing. Because all the wastewater coming out of your homes needs to be directed to go into that STP, And so if funds are the crisis, which that there are, so we need to improve the centralized version and meanwhile look at decentralized treatment options. That means when I say decentralized treatment options, it means that you look at the treatment where it is generated on a smaller scale, so that you treat it so that when it is overflowing into a lake or a river, then it is at least treated water. Even if you're for starters partially treating that water from 0 to 50 is also an improvement. The river, a river rejuvenates itself, but if you're constantly pumping in sewage, how is it going to rejuvenate itself? So you need to treat that water before it goes. So what we look at is decentralized treatment plants. Of course, in NEERI, we are also working on centralized treatment, improving sewage treatment plants, improving effluent treatment plants for the industries. There is a whole section of wastewater technology, but I'm focusing only on what me and my group do in the environmental biotechnology. We address wastewater treatment at a decentralized, option. So when you look at a nala, which carries your untreated sewage. So what are the challenges here? So the challenges is not only the contaminated water, but we have this tendency of dumping all our kachra, into the water. Once it is out of sight, out of mind, तो river में डाल दो, river बहा के ले जाएगा and अपने आसपास तो साफ रहेगा. This is the mentality that somehow or the other we have been brought up with. So when you want to treat, a place like this, you only don't have to look at what is the COD, what is the BOD, what are your pollutant levels, what are suspended solids. You also have to look at the solid waste. So the cost, the operation, the maintenance, everything becomes, really different. So there are different methods to treat these kind of, decentralized options. You have phytoremediation, that is the use of plants. You have constructed wetlands, and you have microbial remediation. So there are merits and demerits of each system, but I won't really go into too much of detail. I would just like to say that when you say phytoremediation, it is of course long lasting. Phytoremediation means the use of plants, but then the time taken, for the plants to grow and for their maintenance, it takes that much of time, which we, actually don't have, because we have already crossed that really threshold. Constructed wetlands are being practiced in many places. The Namami Gange group, the Ministry of Water, they have really done a lot of work across the Ganga basin. They to look at how the catchment areas can be saved, how you can do a filtration kind of through these wetlands so that the water reaching the rivers, is treated. So the only problem here is that it requires a lot of land. The space required around it required, but this is one of the very successful management programs being taken up. And then you have, of course, microbial bioremediation. So the thing with microbial bioremediation is that you need to give that water some contact. You cannot have a flowing water and bacteria and say ki now microbial bioremediation is happening. No, for the bacteria to act, there has to be some kind of holding time. Bacteria to grow itself, and they, in fact, they grow very quickly, more than any other species in the world. In 20 minutes, E. coli doubling time is the doubling time for E. coli. And so it is one of the efficient thing, but of course, it requires that holding time. And you require kind of like a bioreactor kind of a setup. And the recurring cost is there, but maintaining of this bacteria to grow them in that large numbers is also like a challenge. So what we did at NIRI, we made a combination of all these. So what we did was so there was something that came up that interrupted the flow. But what we did at NIRI is this. So these kacha nalas that are there, we first made them into pakka nalas, and we created units in them. So you can see that there are walls being put within this drain, so which increase the holding capacity. So we can use a combination of bacteria and also a combination of plants. So we use species like canna, which are, grow in the sewage water also, and they take up all these nutrients and, which cause eutrophication basically. So it's called Renew, this technology, and it was prepared by a team of engineers, chemists, biologists. And what we did was we created this kind of, kacha drain carrying sewage into something like this. Not only is it aesthetically, good to look at, compared to this, you can walk even around it without any problem, but it also meets the CPCB standards of, water that can be discharged into the river. Now, I was the project leader for this decentralized treatment, which we applied during the Kumbh Mela of 2019. So this is where the drains were, these 6 drains were given to us. and Namami Gange, along with the state mission of Clean Ganga in Uttar Pradesh. They gave us these sites, and you can see it is in the Kumbh area, and all these drains lead to the Sangam point where all the devotees were there. So this was an extremely challenging, but I take great pride in this project because it gave us an opportunity to take our methods, the technology that we built at NIRI, into the field and into national use, So basically what we do is we look at the natural attenuation capacity. When I say natural attenuation, it means the bacteria actually already have the degradative capability. So, but nobody knows of it, When we talk to the engineers about treatment plants, they use the activated sludge process. And for them, the activated sludge is a black box. But actually, that is the component that is causing your entire biodegradation. And so you cannot use it as a black box. We take out the DNA from that. It is called, the metagenome. When you take a single bacteria and look at its DNA, it's called genomics. But when you take the whole community, it is called a metagenome. So we look at what is the inherent taxonomy, what are the kind of bacteria that are there in the metagenome, what is the functional capabilities, what kind of degradative genes are there. We look at what are the stress responses. Sometimes you have the relevant bacteria in the niche, but they are not able to act because they are under different stress. Just like you and me, we cannot function under stress. And similarly, bacteria also living, they also have some stress which they cannot function. So we answer all these questions, and then we try to put a new dimension, into wastewater treatment, which is very different from the conventional, treatment being done. So I just explained, and this is, what we do. And when we study all that, then we can say, what kind of bioaugmentation we require? Do we require X genes or we require Y genes? What kind of stress does the nutrients and the dissolved oxygen play? So how do we improve the efficiency? All this can be answered by looking at metagenomics. Now, this is a picture that was published, in a journal called Environmental Microbiology. It is not my work, and it very beautifully explains the point that I'm trying to show you. So all these are happening in nature, In nature, you don't have one single contaminant. You have so many, all this outside the circle. Suppose these are all the chemicals that you find, in any niche. For them, to lose their toxic potent, they have to be biodegraded. So they are biodegraded through various cycles, through any microbes. Could be fungi, could be bacteria. And then they come to the central metabolism, where the finally mineralization comes. The rings open and they are degraded into carbon dioxide and water, which are, of course, the complete mineralization of those compounds. So if we want to study, normally, I'm sure all of you are teachers, that if you are giving a PhD student a topic, you would take one compound and follow its degradation up to here. So you imagine how many PhDs I need and how much of time I need to evaluate everything singly. So what metagenomics does is it gives me all these answers in one DNA sequencing. A metagenome represents the entire community of that niche. So all the functions that are happening, that answer is given to me. And so which is why we look at, the DNA aspect. This is called the metagenome. So like I said, there is a difference. Don't get confused between genomics and metagenomics. This is a single bacterium and this is a community. So what it gives us is, it tells us what kind of microbial population is there, what is the role of each microorganism in the environment, what is the functional capacity, what are the new genes and new pathways that we don't know. You know that all the microbiologists in this group will know that today only 5% of our microbial diversity is reported. The remaining 95% we don't know. So there could be new pathways, new genes in the environment that we don't know, which metagenome will give you an answer. I don't know how much time I have, but maybe I will very quickly go through some case studies to highlight what metagenomics can do. So we went to a petrochemical complex and we looked at different bacterial taxonomy. Now, both the wastewater from this industry is extremely diverse in nature. It's not the same at all. So we took 4 different time points and we did the metagenome. And of course, you have very different. What we found was when you say bacterial genera, there are majority of them were conserved between these plants, which was really very surprising for us because the wastewater composition is different, but still the genera was same. So basically, we need to look at what is, the functional aspect of it. So what metagenomics can do is it tells you the entire pathway. Now, this I'm only highlighting with phenol. So from phenol, to go into, glycolysis or into the citrate cycle, there are a set of 15 genes that are involved. So through the metagenome, I can say if all these genes are present or absent and how many numbers abundance. So when you have more numbers, you know that these pathways are more operative. So this is something that only metagenomics can do. So that was only phenol. You can extend the same into all of it. So you see these colored squares. If all the 4 colors are present, it means that the gene to convert this compound to this compound is present. So same here, but here when you come, you don't have those genes present. So this is where your your cycle in nature is going to stop. And this is where you need bioaugmentation to continue with this cycle. So this kind of an information is given. We did this exercise for a common effluent treatment plant, in Hyderabad. All these dotted lines are what genes that are present in that effluent treatment plant. And these bold lines are the genes that are not present. So for example, if you say have benzoate, it is easy, of course, toluene, biphenyl, coming in the wastewater, it's going to be degraded and gone out. But if you have phenanthrene, aniline, this system doesn't even have those genes. So those compounds are not going to be degraded and they're just going to flow out with the wastewater. So this is where we told the company that they need to do bioaugmentation of these kind of bacteria. So this is how they improve, their efficiency. So I won't go into all this because I think I'm already taken a lot of time. But so when you do a metagenomic analysis, it gives you all the genes that are, responsible for removal of your BOD, COD in wastewater. This is all the degradative capacity of, the aromatic system. And the plants that we are adding are taking up nitrogen. And so we also looked at nitrogen genes. So, the presence of these, nitrifying and denitrifying genes tells us that the system is working. Otherwise, nitrogen is one of the major causes for eutrophication of lakes. But what was very disturbing is in our analysis, we found a number of multi-drug resistant genes. And you can see that they are so constant from different niches, So this is something that is really worrisome. WHO has also, declared that antibiotic resistant bacteria are one of the now top 10 global health threats facing the population. Tomorrow it's going to hit our heads, like COVID did. And we really, what can you do when your pathogen in your body is not responding to your antibiotics? This is the seriousness of the issue. And all this comes from untreated wastewater because your domestic waste has a lot of antibiotics, right? All of us are using antibiotics. And after COVID, it is more rampant. you have one sneeze and people take erythromycin because you don't want COVID, you know, and which is understandable, of course. There is no, solution. Your health is there. But that water is not being treated. This is what you're not understanding. So unused antibiotic, which is being dumped into the environment, that also is there. So we are seeing a very big increase, which is really very scary. We have looked at multi-drug resistance not only in wastewater. We have looked at all the lakes, in Nagpur. We have looked at it in the Ganga River in different things, and everywhere there is a really huge rise, of this menace. So what your genomics allows is, allows you to show how to turn on a functional gene. Our functional gene is by remediation genes. Okay. So the genomics allows you to see what kind of factors are there. And bioremediation is not really easy. There are these many, all these things that you can see here that are essential for the microbes to actually work and bring about the biodegradation. So this is something that has to be seen. And with all the study, we are able to take a decision on the site whether we need to introduce more bacteria or more genes basically that are required, or are they already there and we only need to do a stimulation. We need to remove the stress factors. We need to probably provide them more food, those kind of decisions that we make. And so if you have a polluted river and if you want to take an acceptable, bring it to the level of an ecological balances, it's not only the researchers. You need participation from industry. They have to take that responsibility to, treat their wastewater before you are putting it in. You need the environmental forums, that coordinate with us. And most important, you need public interaction. Without the public interaction, no amount of treatment is going to work. Because today I set up a decentralized treatment plant and tomorrow the public dumps solid waste into it, dumps their plastic bottles into it, then whatever we have worked for, whatever, the government or the policymakers have invested in to treat this wastewater is not going to work because the public had no respect for that and they have used it to dump their solid waste. So you see, it is not only the responsibility of researchers. Any environmental sustainability program needs to have a combination or an agreement with all these heads that I have written. So if you want to take your environment, this is the scenario today actually. From green, we have already moved into gray. But what we need to do is move the gray back into the green, this is a very overused quote, but it still applies. We say we have not inherited the earth from our ancestors. We have borrowed it from our children. So it is our duty to change this green, gray back into the green so that, our children can still drink water, safe water. So thank you. And I apologize if I have taken, extra time. I'm going to stop sharing. So Dr. Meshram,
I'd like to request all participant, please ask your queries. Have an interaction with our resource person. Dr. Archana Mahakalkar, madam, vichara.
Then, after doing genomics, we come to know the functional genes of that bacteria. So, how further process takes place, ma'am?
how? Sorry? How what?
What next we are doing? with that bacteria?
So when you know that, see, the thing is, why we are doing this metagenomics, to find out what is the function of a bacterial community, if these kind of genes are present or not. And so that can be used for the bioremediation. This is what we are doing. So when we say that the degradative pathway is present in an environment or it is absent. So if we find it is absent, then we do the bioaugmentation. You just don't blindly do a bioaugmentation. You have to know which kind of bacteria you need to put in. So which is why functional gene analysis is very important. So if it is missing, we add it. If it is already present, then we look at what are the stress factors that are preventing this microbial community from carrying out the biodegradation, and we address that.
Okay, got it, ma'am. Thank you.
Thank you.
Ma'am, ma'am, is herbicide harmful for soil fertility?
See, any kind of xenobiotic is going to change your ecosystem. So I would say that the desired quantity, you should not exceed that desired quantity. Of course, if you're putting a chemical and that chemical is killing, say, the grass or the insect or something, that means it is a toxicant. So basic definition is that it is a toxicant. But there are herbicides that have natural biodegradation within the soil. So the thing is to use them in a quantity that does not allow them to cross that critical threshold. So the soil should be able to naturally biodegrade that solution after this is over, after its job is over. So you have to give that much time between crops. So all those kind of things need to be taken into consideration.
Thank you.
Again, anybody would like to ask any queries?
So I think I have come into their lunch time, So
Yes, madam.
probably it is important for everybody to take the break.
Otherwise, I would like to request Dr. Vishal, please propose the vote of thanks for this informative presentation.
Okay, sir.
Dr. Vishal, please.
Good afternoon, everyone. It has been such an honor to be a part of this wonderful session. On behalf of the whole team of UGC HRDC Nagpur University and all participants, I would like to extend my heartfelt gratitude to our eminent guest, Dr. Atya Kapley. Thank you. Uh, Sincere thanks to our course coordinator, Dr. Praveen Meshram sir, for arranging this wonderful session. Thank you to all participants. Thanks one again, one and all. Thank you, sir.
Okay, madam.
Okay, thank you.
Okay, madam. So now I would like to request all the participants, we all will proceed for the lunch. And then again, we'll join the meeting at 2:45.
सर सर
हा, कोणाला
सर
विचारा.
माझा अटेंडन्स नाही लागला सकाळी. मी 11 वाजून 5 मिनिटांनी